So our second talk is also from the Transporter Elucidation Network, specifically the Integrated InTech Center. Dr. Pouria Fattahi is an assistant professor at the University of Georgia in the School of Chemical Materials and Biomedical Engineering. His research focuses on developing human-relevant organoid and organ-on-a-chip platforms to study environmental chemical exposures, barrier function, and maternal-fetal health, with an emphasis on placental and vascular systems. Pouria, come on up.
Good afternoon, everyone. Thank you, Lauren, and thank you, MPRINT, for being here and giving me the opportunity to present our work. So I'm going to talk about the placenta-on-a-chip model in order to study the mechanistic insight of maternal-fetal after exposure to environmental toxicants. So a lot of experts in this class, in this room. So fetus can get exposed to a lot of toxicant chemicals or toxicants or chemicals during pregnancy. It's a variety of ranges: air pollutions, smoking, alcohol consumptions, first-hand or second-hand smoking, as well as the chemical toxicants, including cadmium. So they can get exposed to these chemicals. However, we do have a protective shield naturally that can keep these toxicants away from the fetus. It's called placenta. It is somehow, to some extent, it's difficult to study these toxicants due to a variety of reasons, such as ethical constraint. It's a very complex tissue. It's just constantly changing during the 9 months of pregnancy. There's an individual variability. It's difficult measuring low doses specifically during pregnancy, and also it's a combined exposure unpredictability in human organs. So what is the solution? So the question that I always ask is that how can we study these conditions? And from the talk, title of the talk, you can probably guess. My solution, the direction that my lab and our collaborator will take, is using organ-on-a-chip. So these organ-on-a-chip are tiny devices. It's in the size of flash memory device. The object is that to create an environment that we trick cells so they think they are in their native environment. So there are a couple of elements that we want to achieve: structure, most likely 3D structure, as well as using human cells, and then make the functionality similar to what's happening in the human body. So that's a goal that we want to achieve with these tiny devices. So I typically use this phrase, that conditions beyond reach. So there are difficult conditions such as pregnancy that my lab is interested to study. So when we want to develop a new organs, such as the placenta barrier, so we need to look at the anatomy. So in this case, we are having a barrier that has cells toward maternal blood circulations called syncytial trophoblast, the lining, and also the fetus side has blood vessels. So we want to exactly create this barrier, so small interface between maternal and fetal circulating blood. So with our design, we actually create a multi-compartment chip that has a bottom part and top part. The bottom part, we call it fetal compartment, and the top part, we call it maternal compartment. And we are using specific cells for these two compartments, which I'll explain in detail in the future slides. So this has multi-layer fabrication. The bottom has 3-lane channel, so the bottom compartment. There is a porous membrane with the size of 1 micron pore size membrane in between the top and bottom. And the top compartment has an inlet and access port for the flow that we are introducing. So you can see a cross-section of the real part that are pseudostained for different compartments. The bottom, which is red, the side channels, and then the top, which is colored yellow. During the fabrication, we started with cell lines initially, but during the work, we came up with this amnion foundation, and in 2022, we got this seed grant that provide us an opportunity to work with cells from term placenta. And from amnion foundation, we reach out, we did have a through discussion to be able to get the cells from the primary human tissue So we were able to achieve 4 different cell types: cytotrophoblasts, endothelial cell, fibroblasts, and tissue-specific macrophage half-power cells. And the beauty of working with Amnion Foundation was that we were able to get all of them from the same donor. So it's not only primary cell, it's donor-specific. And then later on, you will see that we also be able to achieve the tissue from the same donor. So imagine one of our chips. So now we are looking at the cross sections. So in these cross sections, we have 3 channels on the bottom compartment. The middle one has a 2 millimeter in size, and the 2 side channels has 1 millimeter. And then these chambers, as I mentioned, they are separated. The chambers are separated with the porous membrane. And then we call the bottom one, again, like fetal compartment or vascular compartment. You will see why. And then we have the maternal chamber on top. First, we are injecting a solution of the primary endothelial cell and fibroblast ECM hydrogel in the middle channel. And the following day, we are adding endothelial cell to the side channel in order to create a perfusible microvascular network. After 7 days, we have a vascular network created in our chip. So the vasculogenesis is happening in 7 days. And then you can see that this has been done in most of our work, most of the time. The vasculogenesis is happening over the course of 3 days. So you see the 2 cell lines that are labeled with GFE and so red label and green label. So the side channels are creating perfusible vessels. And after 7 days, our blood vessel, microengineered blood vessels, can be perfused with beads, with immune cells, circulating cells, and in this case, you're seeing 500 nanometer size fluorescence beads perfusing through these engineered blood vessels. So this is a common practice in our lab. So we do create this engineered vessel for different organs, but the process takes 7 days. And after we are sure that this is functional, we have perfusible vessel, it's time to add a third cell type, which is cytotrophoblast on the top compartment, and do this essentialization process. So again, going back to the fabrication of fetal compartment, for the first 7 days, we are adding endothelial cell and fibroblasts to our microfluidic devices, and we wait 7 days for them to create the vascular network. And as I showed with the beads, these cells, after 7 days of vasculogenesis, these vessels are perfusible. So now, in this time, we added red blood cells. So you can see a video micrography that's showing the red blood cells can be perfused through one single microengineered vessels in our chip. So perfusion is the functionality of our bottom compartment. After we are sure, we are moving forward to the top compartment, adding the cytotrophoblast from Amnion Foundation. We did culture them for 3 days, and they go through spontaneous sensitization. So we see the microvilli formed in our chip after 72 hours significantly compared to the cytotrophoblast. We also see the sensitization process, fusion of the nuclei in our system, as well as measuring beta HCG. We can see a significant increase after 72 hours in our chip. Needless to say that most of this work has been done in low oxygen tension, and there's a significant difference between beta HCG after 72 hours if you're culturing them at low oxygen tension versus normal oxygen tension. So we also look at the RT-PCR expression of different markers related to sensitization, and you can see most of the markers are related, such as the transporter or sensitization marker are highly upregulated, and some of the ECAT here in OZO1 is reduced. And the middle compartment is actually a device without flow. So that also shows that the flow has a significant effect on expression of the genes in our model. So we also look at the transporter, in this case GLUT1 expression in our model after 72 hours. And then our complete model compared to perfusion and non-perfusion system has a glucose transport rate similar to what has been reported in the literature as physiological range. So the model is the most complete compared to the one that we don't have perfusion or we don't have the blood vessels at all. So now it's time to expose this chip that went through extensive characterization in terms of functionality to the cadmium as a target environmental toxicants. So we then introduced this cadmium at the doses that are comparable to what has been reported in the literature as physiological range and beyond that, and compared with the gold standard Transvaal. So our model is more resistance even at higher concentrations. So as you can see in this immunostain images, even at low concentration of 1 micromolar in the Transvaal, we can start seeing some dead cells where we won't see this in our model. So the model is more resistant to cytotoxicity. We measure LDH from the top compartment and bottom compartment. This is something unique to our model, that we are able to separate maternal compartment from the fetal compartment. So they have different inlet and outlet. We are able to collect samples from cytotrophoblasts. As you can see, the maternal compartment. In the dynamic, static, and Transvaal, we measure a different dose of cadmium LDH release. And also, we did the same thing with the fetal compartment, which is the bottom compartment, where we collect it. We look at the caspase activity in both dose-dependent, and for 10 micromolar, which is the highest dose that we aim for, we look at the kinetics. Transport rate also shown that here we were able to measure the transport rate in our model using this chip. So we were able to see the transport rate of cadmium is increased when the cadmium concentration is higher. So another interesting thing is that even though we had low number of macrophages in our hand, we were able to incorporate them in part of the study. So now in this part of the study, we have 4 cell types: endothelial cell, fibroblast, cytotrophoblast, and macrophage, HAFR. And then we measure the cytokine release from both maternal compartment and fetal compartment at a high concentration of 10 micromolar. And what we observe is that in both cases, in all the cytokines that we measured, it's statistically significant release of these cytokines when we have HAFR cells in our system. So that shows that if you're missing these immune cells, your model might not be completely representing the human physiology. We are also able to measure separately maternal compartment and fetal compartment gene expression, a series of gene expression including the transporters at 1 micromolar, which is medium, and 10 micromolar, which is high concentration of cadmium. And you can see that most of these transporters started to have higher expression of their gene in our model. We also look at the caspase activity, some of the markers related to fibrosis in our fetal compartment, where we have the fibroblast, and we see a significant difference when we are increasing the cadmium even in the fetal compartment. And also, I wanted to highlight that the cadmium is only introduced to maternal compartment, circulating maternal compartment. So now this chip has done a lot of endpoints that is traditional. Can we incorporate this to more of an advanced technology such as metabolomics? The answer is yes. In collaboration with Princeton, what we did, we collected the samples from maternal compartment at the dose-dependent exposure of 0.11 and 10 micromolar, and look at the metabolomic profile of different chips in our system. And what we have observed is that in low doses, there is no difference between metabolites, but 184 for the maternal compartment were significantly higher or changed in even low concentration of 10 and 1 and 10. And then from there, we were able to analyze the metabolite pathway metabolomic changes of 10 micromolar, 1 micromolar, and 0.1 micromolar, and compare them together for the maternal compartment. And what is happening is that a lot of these metabolites that are significantly changed are similar between 1 micromolar and 10 micromolar of maternal compartment. And we further were able to identify 25 biomarkers from each metabolite changes in each concentration, and 5 of them are similar between 10 and 1. And the interesting thing for our chip is that we are also able to do the same thing with the fetal compartment. So we did the dose-dependent study of metabolites, 0.1, 1, and 10 in our fetal compartment, and we did the same analysis, compared the pathway that are changed significantly at 10 and 1 micromolar And among all the biomarkers that we were identified, one of them were common between maternal and fetal compartment. So now the question is, how can we validate this? So the validation, we went back to Amnion Foundation and asked them if they can give us the same tissue from the same donor, and luckily they had it in their bank and gave us the placenta explant. So we designed a new chip that we were able to expose them to cadmium over the course of 24 hours, and we kept them 48 hours in the chip, so 72 hours in total. The cells are still viable, so they have the similar function from day 1 to 72 hours, and we did the dose-dependent cadmium study. So we compared all the cytokines that we did in our chip, now in explant, and then put them side by side next to each other. So we have the explant as a validation, maternal compartment, and fetal compartment. And in terms of dose-dependent study of cadmium, you can see that, for instance, IL-1 beta comparison between cadmium exposed explant as well as our fetal compartment and maternal compartment. We also went one step ahead and did the same analysis in terms of metabolomics for our explant system. Dose-dependent 0.1, 1, and 10 macromolar in our explant. And we were able to see significant changes in metabolites and pathways that we analyzed and put them side by side in our microfluidic devices. Now, there are 11 pathways that are similar, but the advantage of our system is that we are able to identify which metabolites are changed in the maternal compartment and which metabolites are changed in fetal compartment. This is something that we are missing in the explant, and now we can compare them and know exactly what is the source of these metabolite changes in explant if you're going through our microfluidic devices. So in conclusion, so the goal here was to show that we are able to create a 3D placenta model with high physiological accuracy. We can assess cadmium in dose-dependent and time-dependent. We were able to look at the BCRP transporter as a protective mechanism, which I did not talk about it today. It's a very big study. It will appear in Nature Biomedical Engineering very soon, so we can discuss about the BCRP if you're interested. And then we were able to uncover some of the potential biomarker by just using metabolomics and our microfluidic devices. So, and again, at the end, I want to highlight that organ-on-a-chip model as a powerful tool for study pregnancy-related environmental health issue. With that, I want to thank our collaborators, specifically Danha and Lauren, and the rest of collaborators. Thank you.
Thank you, Pouria.